Welcome to Roadmap to Residency Evidence-Based Applications. We are starting our immunology module, and in this video, we will be discussing through some of the basic concepts of immunology. I'm Aviraz. Welcome to our Club to 50 Matchmaker sessions. So whenever we start studying immunology, the first concepts that we need to clear out is the difference between the innate immunity and the adaptive immunity. So the innate immunity consists of neutrophils, macrophages, monocytes, dendritic cells, natural killer cells, complement system, physical epithelial barriers, or secreted enzymes. The adaptive immunity consists of T cells, B cells, and circulating antibodies. So the mechanism behind innate immunity is that it is germline encoded, and adaptive immunity is that it is through the variation through the VDJ recombination through the lymphocyte development. So the response to pathogens in case of innate immunity is non-specific, so it occurs rapidly with no memory response. Whereas adaptive immunity is highly specific and it is refined over time. So that's why it develops over long periods, and there is memory response, which is faster and more robust if the similar form of antigen comes in contact with adaptive immunity. So the secreted proteins of innate immunity consist of lysozyme, complement, C-reactive protein, defensins, and cytokines, whereas adaptive immunity consists of immunoglobulins and cytokines. So the key feature in pathogen recognition in innate immunity is the toll-like receptors, whereas in adaptive immunity, we have talked that there is a memory response. So the memory cells, which are the activated B and T cells, are the core function or the core behind the pathogen recognition in case of adaptive immunity. So the adaptive immune defenses have the following characteristics in common. So each B and T lymphocyte is specific for a particular antigen. As population, lymphocytes have extensive diversity, are enhanced with each repeat exposure, so that's why they have immunologic memory and are capable of distinguishing self from non-self, and they are self-limiting. The features of adaptive immunity are designed to give the individual the best possible defense against diseases. So the specificity is required along with immunological memory to protect against persistent or recurrent challenge. Diversity is required to protect against the maximum number of potential pathogens. Specialization of effector function is necessary so that the most effective defense can be mounted against diverse challenges. The ability to distinguish between self-host cells and non-self, that is the pathogens, is vital in inhibiting an autoimmune response. So the self-limitation allows the system to return to a basal resting state after a challenge to conserve energy and resources and to avoid uncontrolled cell proliferation resulting in leukemia or lymphoma. So whenever there is no self-limitation, you get diseases like leukemia or lymphoma. So we also need to get some clear concepts about the major histocompatibility complex. So there are generally two MHC molecules, that is MHC1 and MHC2. So MHC is encoded by HLA genes, and they present antigen fragments to T cells and bind to the T cell receptors. So the loci is HLA-A, HLA-B, HLA-C, and as MCA, while talking about MHC2, the loci is HLA-DP, HLA-DQ, and HLA-DR. So MHC1 have loci one of only 1 liter, while MHC2 have loci of 2 liters. So the binding of MHC1 molecule is through TCR, that is the receptors, and CD8, whereas for MHC2 it is CD4. So you just need to multiply that. 8 times 1 is 8, and 2 times 4 is 8. So you just need number 8. So that's how we remember this. So the MHC1 molecule consists of 1 long chain and 1 short chain, whereas MHC2 consists of 2 equal length chains, that is 2 alpha and 2 beta units. The expression of MHC1 is in the all nucleated cells, the antigen-presenting cells, and the platelets, whereas MHC2 is only in the antigen-presenting cells. The function is to present the endogenous antigens to CD8 plus T cytotoxic T cells by the MHC1 molecule, where it presents to CD4 plus helper T cells. So again, CD8 and CD4 concepts. The associated proteins, which is important here, is that MHC1 have the associated proteins beta-2 microglobulin, and MHC2 have an invariant chain. So talking about the functions of natural killer cells. So natural killer cells are the lymphocyte member of innate immune system. They use porphyrin and granzymes to induce apoptosis of virally infected cells and tumor cells. The activity is enhanced by interleukin-2, interleukin-12, interferon alpha and interferon beta. It is induced to kill when exposed to a non-specific activation signal on target cell and or to an absence of an inhibitory signal such as MHC1 on target cell surface. It also kills via antibody-dependent cell-mediated cytotoxicity. The basic functions of B and T cells. So the B cells, they take part in this humoral immunity. Now this concept is very, very important. So B cells take part in humoral immunity. They recognize and present the antigen and they undergo somatic hypermutation to optimize antigen specificity. They produce antibody and they differentiate into plasma cells to secrete specific immunoglobulins. And they also maintain the immunological memory, that is, the memory B cells persist and accelerate the future response to antigen. The T cells, they take part in the cell-mediated immunity. The CD4 plus T cells help B cells make antibodies and produce cytokines to recruit phagocytes and activate other leukocytes, whereas CD8 plus T cells directly kill virus-infected and tumor cells. So the T cells take part in delayed cell-mediated hypersensitivity type 4. So we have a different chapter on hypersensitivity that we will be dealing on the video. And they also take part in acute and chronic cellular organ rejection. Now, this is the timeline of the immune response to an acute infection. So first is the first, there is innate immune response, and then there is the induction of adaptive response. Okay, first the innate immune response acts whenever there is entry of microorganism, and then the adaptive immune response gets inducted. Okay, it gets stimulated. After that, there is adaptive immune response which takes over, and when the infection or the pathogen is cleared, then there is the immunological memory, so that whenever again the same infection or the same pathogen comes in contact as an antigen, there is a more rapid response by the adaptive immune response. So this is the pathway whenever there is the newest exposure to a very, very new antigen. So, talking about the different interaction between the innate and adaptive immune responses, so there is anatomical and physiological barriers. After that, there is the innate immune response, which consists of phagocytes and granulocytes and the complement systems. Now, through cytokines, there is activation of the adaptive immune response, which consists of T lymphocytes, B lymphocytes, which produces antibodies, okay? And these antibodies, whenever there is again re-exposure, they again reactivate in a super added form, okay? Now, there is even a very a bigger response than the previous one because there is a memory that have been formed towards that specific antigen. So the innate and adaptive immune responses do not act independently of one another. Rather, they work by a positive feedback mechanism. The phagocytic cells recognize pathogens by binding the PAMPs through various pattern recognition receptors, leading to phagocytosis. The phagocytic cells process and present antigen to facilitate the stimulation of specific T lymphocytes, with subsequent release of cytokines that trigger the initiation of specific immune responses. So T lymphocytes produce cytokines that enhance the microbicidal activities of phagocytes. The cytokines released by phagocytes and T lymphocytes will drive the differentiation of B lymphocytes into plasma cells and isotype switching. And then the antibodies will aid in the destruction of pathogen through opsonization, complement activation, and antibody-dependent cellular cytotoxicity. This is the basic format or the basic mechanism through which our body's innate and adaptive immune system acts in corresponse. So which of the following is most likely to cause a faster and stronger immunological response against the same infectious agent after re-exposure? So adaptive immunity and immunological memory, as you all know by now. So the innate and adaptive arms of the immune response work in collaboration to stop an infection. So once a pathogen has broken through the anatomic and physiological barriers, the innate immune response is immediately activated. Oftentimes, it is able to contain and eliminate the infection. Now, when the innate immune response is unable to control the replication of a pathogen, the adaptive immune response is engaged and activated by the innate immune response in an antigen-specific manner. Typically, it takes around 1 to 2 weeks after the primary infection for the adaptive immune response to begin clearance of the infection through the action of effector cells and antibodies. And now, once an infection has been cleared, both the innate and adaptive immune responses cease. Antibodies and residual effector cells continue to provide protective immunity, while the memory cells provide long-term immunological protection from subsequent infection. Thank you for watching. Keep studying hard.